Hello, thank you for coming. I'm Dong-Young Kim, a master course student at Seoul National University. Today I'm going to be talking about some different type of cave dweller. In this conference, I've seen a lot of evidence that many hominins used to live in caves. And actually my research animal are harvestmen or the cave dweller as well, but actually they are quite way smaller and they actually do not have the spine. Looking like this. Okay, so let's talk about this unique animal and their evolutionary history. As it is well known, there are weird animals living in the cave, and these remarkable creatures have captivated many biologists, including Charles Darwin, mainly due to their intuitive and extreme illustration of fundamental evolutionary principle that environmental change drive adaptation leading to the phenotypic change. Then what exactly fuels this dramatic evolution? We can think about the two things. The first thing we can imagine is that cave is very harsh environment. Such conditions impose an intensive selection pressure on organism. Secondly, the isolation of the cave ecosystem accelerates the evolutionary processes. These two features interact, resulting in profound evolutionary changes in cave animals. However, in many studies of the cave animals until now, these features have often been considered separately. Many macrobiologists have often focused on some neutral evolutionary patterns of this isolated ecosystem, while many experimental studies concentrated more on some genetic and developmental mechanism of the adaptation in cave. I gotta say that these both of approaches are undoubtedly insightful and inspiring, but in order to deepen our understanding of cave animal evolution, more integrative framework continuously encompassing both neutral and adaptive evolution is needed. In our study, we tried to fill the gap by taking some integrative study in whole genome scale to an intriguing model system, the genus Kaolinonychus. It is an invertebrate belonging to the order Opiliones, which is also known as harvestmen or daddy longleg, related to spider, mite, or scorpion. They really resemble the spider, but they are actually not a spider, differ in having their fused body structure and lack of the ability to produce the venom or silk. Within Opiliones, here the jelly-like adorable harvestman endemic to Korea is genus Kaolinonychus. It consists of a single species with two subspecies, the coreanus and longipes. Please remember these two subspecies names. Then why this lesser known animal, ideal model system for studying the cave animal evolution in an integrated manner? First, they include both cave and surface population, making some comparative feasible. And as they lie at the boundary of the species, we can observe the evolution following the transition into the cave. And their lifestyle and dispersal patterns are quite simple and predictable, making the evolutionary model more straightforward. And the availability of the sample and manageable genome size make them a suitable model for whole genome study. Based on this consideration, we selected Kaolinonychus for a model for studying the cave animal evolution. So our goal is to reconstruct the evolutionary history of this species, which we divide into three main phases. First, producing our genome of this species to lay the foundation of a whole genome analysis, and then performing the population study to uncover the demographic history. Using this information, we aim to properly identify the adaptive signal in cave population. To this end, I collected around 400 individuals from both surface and cave population across the Korea. And then selected 188 individuals and sequenced in whole genome scale. Okay, I'll skip some methodological details here. The first major result of us is that we successfully made the first reference genome of this species and we utilized it into our downstream analysis. So let's move on to the population structure analysis. This PCA is visualizing the genetic similarity among individuals. We could identify 6 major genetic clusters and mark their distribution onto this map. The first thing I want to note is that these Longipes and coreanus, the previously described 2 subspecies, clearly separate along the PC2. And now let me briefly introduce the main character of this study. The first one is this Longipes, named for their relatively longer walking leg, and they're geographically distributed in central and southern part of Korea. On PCA, they extended below the PC2 axis. And the other subspecies, coreanus, is geographically widespread, but in PCA they are clustering at one end with a subtle genetic decline along the PC1 and PC2, forming 3 subgroups. Coreanus Gangwon karst region in green, and coreanus northern in navy, and coreanus cyan in southern, southern in cyan. And for environmental group, the cave and surface populations are marked as different symbol. It is showing that this kind of environmental grouping does not mainly explain the genetic structure in the Kaolinonychus, which implies that the cave adaptation might have been happened independently in each of subspecies. And next is the star of this presentation, previously unknown cluster with a highly distinctive genetic profile. This cluster is confined to a limited area within like a 15-kilometer radius around the mountain Gariwangsan. The genetic difference between the Gariwangsan and the others account for the largest portion of overall genetic variation within Kaolinonychus. And it appears to be the common outgroup of all other members in Kaolinonychus. Okay, so in this PCA space, the relative position of each population help us formulate some meaningful evolutionary scenarios or hypotheses. To do so, it is worth to focus on some intermediate profile in the PCA. This kind of intermediate profile actually can be shaped by different evolutionary scenario. One possibility is zero boundary effect with genetic drift, like this in stepping stone fashion. This does not require some admixture event. On the other hand, one another is the admixture between 2 wholly diverse populations. Okay, given this, let's go back to our PCA and please note the location of population along the PC1 axis. We can find that many of population are located between Orange Gariwangsan and Cyan coreanus southern. So from this, we can make some different evolutionary scenarios that stepping stone manner one to one another, or the mixture between 2 extreme genetic profile. And this evolutionary hypothesis can be statistically tested using F statistics. And this result is showing that the population between Gariwangsan and coreanus were indeed formed by a mixture event between these 2 extreme populations. And there were differences between population in the composition of genetic Gariwangsan. Given that Gariwangsan is common outgroup of all other members, it is somewhat similar to Neanderthals and or modern humans. Anyway, so how much of the genetic composition from Gariwangsan each population has explained the large portion of genetic diversity within Kaolinonychus. So now, in this geographic map, I represent a genetic affinity from Gariwangsan. And given that the genetic affinity from Gariwangsan does not necessarily mean that there were some direct gene flow from Gariwangsan, then we can make some two different hypotheses of generative process of this distribution. Did Gariwangsan directly contribute to multiple populations, or did some population only got some genetic input from Gariwangsan and the others gained this composition of Gariwangsan through some subsequent indirect admixture? And these two scenarios result in the different structure of population graph. By modeling the population graph, we found one graph best explained our data. And this graph is saying that although this is not a perfect fit to scenario 2, but it leaned toward to the scenario 2. So multiple and indirect nested admixture event led to some indirect and complex evolutionary history in this taxon. Lastly, we explored the net signal of natural selection. We focused on the coreanus group as they include numerous cave and surface population. First, we divided them into two environmental groups, cave and surface. And then what we want to do is identify some genomic region evolving abnormally rapid. And because the rapid evolutionary rate in cave population means that there was a strong positive selection under the cave environment. And we could find some signals. These peaks indicate they were the candidate genes of selective gene, and now I'm in the process to match the biological function of these genes. So until this, I can wrap up my result. And I want to point out some significance of my study. Some people can think that this is not a special case because there were some gene flow between populations or similar species, but actually they were very highly divergent group. The genetic distance between Gariwangsan and coreanus and longipes were it's about like 3%, and given that Neanderthals and humans and chimps, gorilla, and gibbons is similar to human and gibbons. So I already said that it's similar to Neanderthal and modern human, but it is actually very as surprising as the admixture integration between human and gibbons. And especially in this kind of invertebrate, many studies consider they do not have some multiple and complex history like this. So it can give an impression in the further study of many invertebrate evolutionary study. And okay, I'll skip this part. And as I showed in the previous slide, I reconstruct the best graph of their relationship, but this is actually not the final goal of this study. What we want to do is reconstruct a biological history by doing some explicit modeling, because this graph still does not have the information of actual time axis and effective population size. After doing explicit modeling, we can match each of some actual geological or climatic events can match it with their evolution. Especially, we found one interesting pattern. They do not show some isolation by distance, but high genetic diversity was shown in very low temperature region. And we can think that there were a refugee region and high altitude, and going to low altitude and low latitude, the genetic diversity is going lower. So it is still a hypothesis in the process, but from this we can think about some many kind of quaternary climate change and response of the montane biota. So So now using the explicit modeling, I'm gonna delve into this pattern and try to find the process. Thank you for listening.